Welcome back friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding cloning vectors from biotechnology principles and processes in detail. So as we all know that specific vectors like plasmids and as well as bacteriophages, they are having higher capability to replicate within the bacterial cells independent of the chromosomal DNA. So the reason why here these specific vectors they are termed as autonomously replicating circular DNA molecules, because these specific vectors they are capable to replicate within the bacterial cells independent of the chromosomal DNA. Is it clear? So let me write it on the board. Specific vectors like plasmids and bacteriophages are having higher capability to replicate within the bacterial cells independent of the chromosomal DNA. So the reason why here these specific vectors they are termed as autonomously replicating circular DNA molecules. So in general, the copy number of the bacteriophage it remains significantly higher within the bacterial cell. So the reason why here these specific bacteriophages they are capable to replicate or they are capable to manipulate high copy number within the bacterial cell. Is it clear? The copy number of bacteriophage remains significantly higher in order to produce high copy number within the bacterial cell. And here, likewise the copy number of the bacteriophage, even the copy number of the plasmid, it remains significantly higher within the bacterial cell. On average, one specific bacterial cell, it may contain 2 plasmids per cell. And even the copy number of the plasmid, it may range around 15 to 100 copies per cell, and even the number, it may increase higher. Is it clear? On average, one specific bacterial cell, it may contain 2 copies of plasmids, and even the copy number of the plasmids it may range around 15 to 100 copies per cell, and even the number of the plasmid it may increase higher. The plasmid copy number of plasmids may range around 2 copies per cell, and even the copy number may range around 15 to 100 copies per cell. 15 to 100 copies per cell, and even it may increase higher. it may increase higher. May increase higher. And here, one of the unique remarkable feature of these specific vectors, here it is nothing but once when we insert a foreign desirable gene within the vector DNA, likewise the other genes of the vector DNA, even the foreign desirable gene, it is capable to replicate within the bacterial cell. Is it clear? Once when we insert a foreign desirable gene into the vector DNA, likewise the other genes of the vector DNA, even the foreign desirable gene, it also undergoes successful expression within the bacterial cell. And here this specific mechanism or this specific process, here it is termed as gene cloning or gene transfer. Is it clear? Once when we insert foreign desirable gene into the vector DNA, the foreign desirable gene is capable to express within the bacterial cell. Likewise, the other genes of the vector DNA, and here the specific mechanism, here it is termed as gene cloning or gene transfer. And here the specific vectors which are most commonly used in recent times, they are constructed or they are designed in such a way that easily we can recognize the specific vectors which are most commonly used in recent times. They are constructed or they are designed in such a way that easily we can recognize the transformants, and here we can differentiate the recombinants from the non-recombinants. So here the specific vectors, they are most commonly, they are accurate in producing the copy number. Is it clear? The specific vectors which are most commonly used are constructed in such a way that easily we can recognize the transformants and we can differentiate the recombinants from non-recombinants, right? Non-recombinants. And if we consider one specific vector, it has some basic features. So here all these specific features, they play an important role for regulating cloning. Is it clear? If we consider one specific vector, it has some certain features. So here, what are the various features which are a part of the cloning vector here? The various features which are a part of the cloning vector, it may include origin of replication, selectable marker, and cloning sites. So here all these are the various specific features which play an important role for regulating cloning. Is it clear? So let me erase the things here. So here, as already mentioned, one specific vector, it may have some of the following features. So here all these specific features, they play an important role for regulating cloning within the vector. So here what are the various features which play an important role for regulating cloning within the vector here? The specific features, they may include origin of replication, selectable marker and cloning sites. Is it clear? So let me write it on the board. Some of the following features that are required for cloning, for regulating cloning may include origin of replication, selectable marker, and the other one is cloning sites. Is it clear? So at first we will be studying regarding origin of replication. So here what is origin of replication? Origin of replication, it is the specific desirable region within the vector DNA which is responsible for initiating the replication mechanism. Is it clear? Origin of replication, it is the specialized region within the vector which is responsible for initiating the replication mechanism. That means in what way? In order to initiate the replication of the chromosomal DNA, here the origin of replication, it plays an important role. In the same way, in order to initiate the replication of the specific vector, here origin of replication, it plays an important role. Is it clear? So let me write it on the board. Origin of replication it is a specific region which is responsible for initiating the replication of the vector, and here it is termed as origin of replication. So here in order to acquire or here in order to obtain the high copy number of the foreign desirable gene, here origin of replication, it plays an important role. The foreign desirable gene which successfully integrates into the vector, the specific foreign desirable gene, it has to remain in association with origin of replication. So here in order to obtain the high copy number of the foreign desirable gene, origin of replication, it plays an important role. Origin of replication is responsible for obtaining high copy number of the foreign desirable gene. So here the foreign desirable gene which successfully integrates into the vector, the specific foreign desirable gene, it has to remain in association with origin of replication. So here origin of replication, it is responsible for regulating the higher production of or for regulating high production of the copy number of the foreign desirable gene. Is it clear? And then after we will be studying regarding selectable marker. So here what is selectable marker? So let me arrange the things here. So here, as already mentioned, in addition to origin of replication, even the specific vector DNA, it has one more other specific region which is responsible for recognizing the transformants, and even it is also responsible for eliminating the non-transformants, and here it is termed as selectable marker region. Is it clear? selectable marker region, it is the specific region which is responsible for recognizing the transformants, and even it is also responsible for eliminating the non-transformants, and here it is termed as selectable marker region. So let me write it here. Selectable marker. It is the specific region. It is the specific region which is responsible for recognizing the transformants and even it is also responsible for eliminating the non-transformants. And here it is termed as selectable marker region. So here most commonly the specific selectable marker region, it is responsible for encoding antibiotic resistant genes. So here what are the various antibiotic resistant genes which are most commonly observed in case of the selectable marker region? Here the various antibiotic resistant genes, they may include chloramphenicol resistance, ampicillin resistance and tetracycline resistance. Is it clear? The specific selectable marker region, it is most commonly responsible for encoding antibiotic resistant genes, and these specific antibiotic resistant genes, they may include chloramphenicol resistance, ampicillin resistance and tetracycline resistance. Is it clear? So let me write it here. Selectable marker region is responsible for encoding antibiotic resistant genes like chloramphenicol resistance, ampicillin resistance and tetracycline resistance. So here what is the meaning of antibiotic resistant genes here? That means in the presence of various antibiotics like chloramphenicol, ampicillin and tetracycline, the specific E. coli cell, it is unable to perform many of the activities because the specific E. coli cell, it is very sensitive to these specific antibiotics. But here, because of the presence of a specific unique feature termed as antibiotic resistant gene within the particular vector DNA, the specific E. coli host which is in association with the specific antibiotic resistant gene containing plasmid DNA, it is capable to resist the effect of these antibiotics. Is it clear? In general, the specific E. coli cell, it is very sensitive to some of the antibiotics like chloramphenicol, ampicillin and tetracycline. In the presence of these antibiotics, the specific E. coli cell, it is unable to perform many of its activities. But here, because of the presence of unique feature termed as antibiotic resistant gene within the specific vector DNA, here the specific E. coli cell which is in association with the specific antibiotic resistant gene containing plasmid, it is capable to resist the effect of these antibiotics, and here it is termed as antibiotic resistant genes. Is it clear? Because of the presence of antibiotic resistant genes, the specific E. coli which has which has the plasmid DNA, it is capable to resist the activity of antibiotics. And here it is termed as antibiotic resistant genes. Is it clear? And the next specific feature which is a part of the cloning vector, it may include cloning sites. So here what is cloning site? So let me erase the things here. So here, in addition to the selectable marker region, even the specific vector DNA, it has one more other specific region which is responsible for inserting the foreign desirable gene, and here it is termed as cloning sites. So here cloning sites, they play an important role for the successful insertion of the foreign desirable gene. And here all the cloning sites, they are very specific to the activity of one consent restriction enzyme. Is it clear? Cloning sites. These cloning sites are responsible for inserting the foreign desirable gene. So here, for the successful insertion of the foreign desirable gene into the selectable marker region, here the specific cloning sites, they play an important role. And here each and every cloning site, it is very specific to the activity of one consent restriction enzyme. So here these are nothing but what restriction enzymes, PST1, PVU1. So here PST1 restriction enzyme, always it is very specific to the consent cloning site. PVU1 restriction enzyme, always it is very specific to the consent cloning site. No specific restriction enzyme within the selectable marker region, it has multiple cloning sites. That means PST1 restriction enzyme, always it recognizes only at this particular location. The specific PST1 restriction enzyme, it is unable to recognize at any other location within the consent selectable marker region. If one specific restriction enzyme, if it has multiple cloning sites, under such condition, it may lead to the complication for the gene cloning. Is it clear? If one specific restriction enzyme, if it has multiple cloning sites, it may lead to the complication for gene cloning. So the reason why each and every restriction enzyme, it has only one specific cloning site. No specific restriction enzyme, it has multiple cloning sites. If the specific PST1 restriction enzyme, if it recognizes at this particular location, and even the specific PST1 restriction enzyme, even if it cleaves at other specific region within the selectable marker region, under such condition, it may lead to the shortage of the specific vector DNA, and even it is also responsible for the occurrence of the complication in the gene cloning. So the reason why no specific restriction enzyme, it has multiple cloning sites. Is it clear? So let me write it on the board. All the selectable marker regions has specific cloning sites which are very specific to the activity of one restriction enzyme. And here no restriction enzyme, it has multiple cloning sites. So let me write it here. No restriction enzyme has multiple cloning sites. So here presence of multiple cloning sites, it may lead to the causes of complications in case of the gene cloning. So the reason why no restriction enzyme, it has multiple cloning sites. And here, in order to recognize the transformants and even in order to differentiate the recombinants from the non-recombinants in general, here we can take the advantage of two specific procedures, and these may include plating method and the other one is blue-white screening. So here by taking the advantage of plating method and blue-white screening, here we can recognize the transformants and even we can differentiate the recombinants from the non-recombinants. Is it clear? So let me write it here. Plating method. Plating method. And the other one is blue-white screening. Blue-white screening. Blue-white screening. So at first we will be studying regarding plating method. So here what is plating method? Is it clear? So let me arrange the things here. So here, as already mentioned, in order to recognize the transformants and even in order to differentiate the recombinants from the non-recombinants, here we can take the advantage of two specific procedures which may include plating method and the other one is blue-white screening. So here in order to understand the procedure of plating method, here we can take the advantage of one of the common example which has mentioned in your NCERT. So if we consider the specific tetracycline selectable marker region, so here the tetracycline selectable marker region, it has two specific cloning sites which are very specific to the activity of restriction enzymes like BAM-H1 and SAL1. So here by taking the advantage of BAM-H1 restriction enzyme, here we can cleave the specific cloning site. So here after cleaving the specific cloning site by taking the advantage of BAM-H1 restriction enzyme, here we can insert the foreign desirable gene. So once when the foreign desirable gene, once when it has successfully inserted into the specific cloning site, now here the specific plasmid, it has become recombinant plasmid. So here after the successful formation of recombinant plasmid, now here the specific recombinant plasmid, it is capable to allow or it is allowed to insert into the E. coli host. Is it clear? After the successful formation of recombinant plasmid, now here the specific recombinant plasmid, it is allowed to insert into the E. coli host. Now here the specific recombinant plasmid containing E. coli host, it is allowed to grow in case of the tetracycline containing medium. Is it clear? So let me show it here. So here it is nothing but what? Recombinant plasmid containing E. coli host. Recombinant plasmid containing E. coli host. So here the specific recombinant plasmid containing E. coli host, it is allowed to grow in case of the tetracycline containing medium. So here it is nothing but what? Tetracycline containing medium. Tetracycline containing medium. Tetracycline containing medium. So here once when the recombinant plasmid containing E. coli host, once when it is allowed to grow in case of the tetracycline containing medium, under such condition, only the ampicillin resistant selectable marker region, it is capable to undergo expression, but whereas tetracycline selectable marker region, it won't undergo expression in case of the tetracycline containing medium. Is it clear? Once when the recombinant plasmid containing E. coli host, when it is allowed to grow in case of the tetracycline containing medium, under such condition, only the ampicillin resistant selectable marker region, it is capable to undergo expression, but whereas tetracycline resistant selectable marker region, it won't undergo expression in case of the tetracycline containing medium, because here the specific tetracycline selectable marker region, it had undergone insertional inactivation because of the insertion of foreign desirable gene. Because of the insertion of foreign desirable gene in case of the tetracycline selectable marker region, the specific tetracycline selectable marker region, it had undergone insertional inactivation. Is it clear? So let me write it here. Ampicillin resistance selectable marker region undergoes expression, but whereas tetracycline resistant gene undergoes insertional inactivation. Now here the specific recombinant plasmid containing E. coli host, it is allowed to grow in case of the ampicillin containing medium. So here once when the recombinant plasmid containing E. coli host, once when it is allowed to grow in case of the ampicillin containing medium, under such condition both the selectable marker region, they are capable to undergo expression. Is it clear? So let me show it here. So let us assume it as the ampicillin containing medium. So here once when the recombinant plasmid containing E. coli host, when it is allowed to grow in case of the ampicillin containing medium, under such condition both the selectable marker regions, they are capable to undergo expression. But here in the first strategy, when the recombinant plasmid containing E. coli host, when it is allowed to grow in case of the tetracycline containing medium, under such condition only the ampicillin resistant selectable marker region, it had undergone expression. But whereas the tetracycline resistant selectable marker region, it had undergone insertional inactivation because of the insertion of foreign desirable gene. Is it clear? But here in the second strategy, when the recombinant plasmid containing E. coli host, when it is allowed to grow in case of the ampicillin containing medium, under such condition both the selectable marker regions, they are capable to undergo expression. But whereas in the first strategy, when the recombinant plasmid containing E. coli host, when it is allowed to grow in case of the tetracycline containing medium, under such condition only the ampicillin resistant selectable marker region, it is capable to undergo expression. But whereas the tetracycline resistant selectable marker region, it had undergone insertional inactivation. Though the specific tetracycline resistant gene, it had undergone insertional inactivation because of the insertion of foreign desirable gene, but here we can differentiate it from the non-recombinant one. Is it clear? In the first strategy, the specific tetracycline containing selectable marker region, though it had not undergone activation or though it had not undergone expression because of the insertional inactivation, but here we can differentiate it from the non-recombinant one. In the third strategy, when the specific non-recombinant plasmid, when it is allowed to grow both in case of the ampicillin containing medium and tetracycline containing medium, in both of the mediums the specific non-recombinant plasmid, it is capable to undergo expression. When the specific non-recombinant plasmid, when it is allowed to grow in case of the ampicillin containing medium and tetracycline containing medium, in both of the mediums the specific non-recombinant plasmid, it is capable to undergo expression. So here both the specific selectable marker region, they are capable to undergo expression in both of the antibiotic medium because here no specific foreign desirable gene, it has successfully inserted. So here because of the lack of insertional inactivation, both the selectable marker regions, they are capable to undergo expression in both of the antibiotic mediums. Is it clear? So let me write it here. Non-recombinant plasmid containing E. coli host when it is allowed to grow in case of both of the mediums, both of the antibiotic mediums, under such condition both the selectable marker regions like ampicillin resistance and tetracycline resistance, they are capable to undergo expression because of the lack of insertional inactivation. So here, one of the specific selectable marker region, it is responsible for identifying the transformants, but whereas the other one is responsible for differentiating the recombinants from the non-recombinants. Is it clear? One of the specific selectable marker region which is having the antibiotic resistant gene, it is responsible for identifying the transformants, but whereas the other specific selectable marker region which is having the antibiotic resistant gene, it is responsible for differentiating the recombinant one from the non-recombinant one. Is it clear? And then after, here we will be studying regarding blue-white screening. So here, what is blue-white screening? So let me arrange the things here. And then after, we will be studying regarding blue-white screening. So here, by taking the advantage of plating method, though we can recognize the transformants, but here, in case of the plating method, here we need to place the recombinant plasmid containing E. coli host in case of both of the antibiotic containing medium simultaneously. So here, placing the specific recombinant plasmid containing E. coli host in case of both of the antibiotic containing medium simultaneously, it is quite cumbersome and even it is little bit clumsy. So the reason why, instead of taking the advantage of plating method, here we can take the advantage of one more other alternative strategy through which we can recognize the transformants and even we can differentiate the recombinant one from the non-recombinant one. And here it is nothing but what? Blue-white screening. So here, in order to understand the blue-white screening, here we need to take the advantage of one specific gene which is nothing but beta-galactosidase gene. Is it clear? So let me write it here. Blue-white screening. So initially, here we need to discard one specific selectable marker region like tetracycline selectable marker region. So here, after discarding the specific tetracycline resistant selectable marker region, here we need to place a specific gene like beta-galactosidase gene. So here, once when the specific beta-galactosidase gene, when it undergoes expression, it is capable to synthesize a specific enzyme like beta-galactosidase. So here, as we all know, each and every enzyme, they are very specific to one concerned substrate. So here, once when the specific enzyme, once when it successfully binds to one specific substrate, it leads to the formation of enzyme substrate complex. Enzyme substrate complex, once when it undergoes some specific conformation changes, from the enzyme substrate complex, enzyme product complex is obtained. And here, after the successful formation of enzyme product complex, the specific product which has successfully formed, it successfully dissociates here. Now here we need to apply the consent strategy. To the specific beta-galactosidase enzyme, once when we provide the chromogenic substrate. So here, by taking the advantage of chromogenic substrate, the specific beta-galactosidase enzyme, it is capable to regulate the production of blue color colonies. Is it clear? Beta-galactosidase enzyme. So here, the beta-galactosidase enzyme, by taking the advantage of chromogenic substrate. So here, what is the meaning of chromogenic substrate here? That means it is a colorful substrate. So here, by taking the advantage of chromogenic substrate or colorful substrate, here the beta-galactosidase enzyme, it is capable to regulate the production of blue color colonies. Is it clear? Blue color colonies. So here, formation of blue color colonies, it indirectly suggests or it indirectly says that the beta-galactosidase enzyme, it had not undergone insertional inactivation. Because of not inserting any foreign desirable gene, in case of the beta-galactosidase enzyme, the specific beta-galactosidase enzyme, it is capable to utilize the chromogenic substrate and it is capable to regulate the production of blue color colonies. Formation of blue color colonies, it indirectly suggests or it indirectly says that here no foreign desirable gene it has successfully inserted. So the reason why formation of blue color colonies, it acts as an indication for non-recombinant one. Is it clear? Non-recombinant. Then in the second strategy, once when the foreign desirable gene, once when it has successfully inserted into the beta-galactosidase enzyme, under such condition, the specific beta-galactosidase, it undergoes insertional inactivation, and even the beta-galactosidase, it is unable to utilize the chromogenic substrate. So the reason why in the second strategy, instead of producing blue color colonies, here colorless colonies or white colonies are formed. So here formation of colorless colonies or white color colonies, it acts as an indication for recombinant one. Is it clear? Beta-galactosidase when it undergoes insertional inactivation under such condition, colorless colonies or white colonies are produced. Is it clear? So here this is regarding blue-white screening. Is it clear? And then after we will be studying regarding vectors which are most commonly used for regulating gene cloning in plants and animals in detail.